everyone. I'm Elizabeth McKenna, Executive Editor of Cancer Discovery. For this OncoAlert academic recap, I'm pleased to present some clinical studies recently published in the journal. Next up is CD20 bispecific chimeric antigen receptor in naive or memory T cells for the treatment of relapsed or refractory non-Hodgkin lymphoma by Sarah Larson, Yvonne Chen, and colleagues. Because multiple trials have shown that relapse on CD19 CAR T cell therapy is associated with antigen escape, T cell exhaustion, and lack of CAR T cell persistence, whereas patients with durable response to therapy have relatively elevated levels of naive and memory T cells, these authors hypothesize that engineering naive and memory T cells to express a molecularly optimized CD19, CD20 bispecific CAR would yield a therapy with a strong efficacy, improved CAR T cell persistence, and reduced CAR T cell exhaustion. A first-in-human dose escalation phase 1 trial evaluating this CAR T cell therapy in adults with relapsed or refractory non-Hodgkin lymphoma showed it was safe with no neurotoxicity or greater than grade 1 cytokine release syndrome, and demonstrated strong efficacy with a 90% overall response rate and 70% complete response rate. And lastly, abatacept plus ruxolitinib and screening for concomitant respiratory muscle failure to mitigate fatality of immune checkpoint inhibitor myocarditis by Joe Eli Salem and colleagues. Immune checkpoint inhibitors can induce immune-related adverse events, including myocarditis, which is rare but has a high mortality rate. In preclinical models, CTLA-4 signaling appears to play a critical role, with the CTLA-4 immunoglobulin fusion protein abatacept attenuating myocardial inflammation. Preliminary reports also suggest that abatacept efficacy can be enhanced by the addition of the JAK inhibitor ruxolitinib, which impairs T cell activation. The authors report results from a cohort of 40 patients with severe immune checkpoint inhibitor myocarditis. The first 10 were treated with current guidelines, and the subsequent 30 received systematic screening for respiratory muscle involvement coupled with active ventilation and treatment using ruxolitinib and abatacept. The myotoxicity-related fatality rate was 60% in the 10 patients receiving standard treatment versus 3.4% in the 30 patients receiving abatacept plus ruxolitinib with respiratory muscle failure screening.